Hello everyone, can you hear me? Hello everyone, can you hear me? Can somebody please send me a message so I know that you can hear me? One second. Good evening, Tanish Sharma. Hi everyone. Just let me know, can you so hi everybody, I'm Dr. Nandini and I am your new faculty for micro. I'm taking these live sessions for you. So So the topic for today, before that, I just want to introduce you to the microbiology world. So I am Dr. Devyani Puri and I am a microbiologist. And I'm going to take your subsequent lectures and try to make this topic of micro easy for you. Right. So how are we going to go about here? So I will be looking here and there because I'm trying to see your messages also in the comment section. Okay. So how are we going to go about here? We will be first starting with the general microbiology, and these are the subsequent topics that we are going to be discussing as we go further. So the first topic for today that we are going to discuss is about our scientists of microbiology. So our scientists are very, very important. And if you are a second year prof student, micro is obviously your subject. So this topic is important because you get a lot of short notes on this topic, right? So you get a lot of short notes on this topic, and if you are a post intern or an intern batch who's trying to crack your NEET PG, so there are a lot of MCQs also that come from these topics. So particularly this topic as well. So we will be discussing it as we go ahead. Um, okay. Great. So the first topic is our scientist, and the first scientist that we are going to talk about is Anthony Phillips von Leeuwenhoek. Right. So I'm also going to try and tell you how you can remember these photos of the scientists because these photos also come as a spotter in your exams, and you just have to match the photo with the scientist's name. Right. So Anthony Phillips von Leeuwenhoek, he has such a long name, right? And he has long hair. So that is how you're going to remember this scientist. He has a long name, he has long hair, right? So, what are the achievements of his? So, can you see the word Phillips in his name? So, Phillips, as we know, makes a lot of electrical equipment, right? So, Phillips also makes microscopes, right? So he was actually the one, Anthony Phillips von Leeuwenhoek, who invented the microscope, right? So this is his first achievement that he invented the microscope. And what he did was he saw small animals under it, right? And he used to call them little animalcules. That means small animals meaning little animalcules, right? Uh, So the next thing, his achievement that he did was he's known as the father of microbiology. Why is he known as the father of microbiology? Because he invented the microscope. Without the microscope, there would have been no microbiology, right? How would we ever able to see small pathogens without the help of a microscope? So that is how you're going to remember him. Microbiology, microscope, right? Phillips, the name Phillips in his word, the word Phillips in his name, sorry. And Phillips makes microscopes. He invented the microscope, and that is why he's known as the father of microbiology, right? So this is the first scientist that we are going to talk about. Next, let's come to Louis Pasteur. So this is how Louis Pasteur looks like. Please remember the photographs because you can get questions based on this. Uh, Okay, so next comes the Louis Pasteur. So Louis Pasteur has also had a lot of achievements, and this is a short note for all the second year professional students who will be giving their professional examinations. This scientist can come as a short note, right? So Louis Pasteur. So let's remember it like this. His name has Pasteur, so he let's start with that. So he is the one that actually formed the Pasteur Institute in Paris, right? So that is the first Pasteur formed the Pasteur Institute in Paris. So in his Pasteur Institute, he was doing a lot of modern, research and everything. Hence, he is also known as the father of modern microbiology, right? He is also known as the father of modern microbiology. Remember it like this, he was doing a lot of modern researches in his Pasteur Institute that is there in Paris. So he's also known as the father of modern microbiology, and this can come as an MCQ. Who is known as the father of modern microbiology? It is Louis Pasteur, right? Then remember L and P, from L from Lewis and P from Pasteur, right? L, what did he do? He gave us the concept of liquid media. So we all know that liquid medias are used for culturing the organisms, right? So who was the one who gave us the concept of liquid media? It was Louis Pasteur. So L from his name, L from Louis, L for liquid media. Then P. P, what did he do? He gave us another concept of pasteurization, right? Just like his name, right? So he gave us the concept of pasteurization. What is pasteurization? So what is actually pasteurization? Pasteurization is a process of sterilization that we do, is a process of sterilization of milk. So we all know there are a lot of pathogens that are actually spread through milk, right? So we do need to do certain kind of sterilization of the milk. So what is that process called? Pasteurization, right? So Louis Pasteur was the one who gave us the concept of pasteurization, right? Now remember another thing about Louis Pasteur. Louis Pasteur had a car called the G Wagon, right? So Louis Pasteur used to go to his Pasteur Institute in his car, which was a G Wagon. A G Wagon is a car, right? So what do we want to know with this? So what are we trying to know? This car. So he actually gave us three vaccines. So he gave us three vaccines which are called as car. Which ones are those? Can you tell me? Yes, I'm seeing a lot of comments. Yes. So the first one is your cholera. The second one is your anthrax. And the third one is your rabies vaccine, right? So he used to have a car, and then he gave the name of three vaccines called as the cholera vaccine, anthrax, and rabies. What was he doing with his G Wagon? Right. So with his G Wagon, he actually gave us the theory of the germ theory of disease. Right? Understood? Is it clear to everyone? So let's remember Louis Pasteur again. Louis Pasteur is he formed the Pasteur Institute in Paris. Yes, I'm seeing a lot of comments. Good. Great. So, from the Pasteur Institute of Paris. He did a lot of modern research, hence he's known as the father of modern microbiology. Then L and P, L for the liquid media that he gave us for the culture of microorganisms. P for pasteurization, just like his name, pasteurization of milk. Then remember, he used to drive a G wagon, right? He used to drive a G wagon car. What was he doing with the car? He was actually inventing three vaccines. So, what all vaccines? It was the cholera vaccine, the anthrax vaccine, and the rabies vaccine. And which car was he driving? He was driving a G wagon, right? And G for the germ theory of the disease. Got it? Is it clear to everyone? Great. So let's come to the next scientist that we have, which is Joseph Lister, right? Joseph Lister. What did he do? He actually, just remember SEP. SEP is septic, right? So what did he do? He actually made an antiseptic, right? He made an antiseptic in that time. So during that time, there were not many antiseptics that were there, right? So he was actually the one that made an antiseptic known as carbolic acid, and he used this carbolic acid to sterilize instruments and wounds of patients, right? So he actually used this antiseptic, which is carbolic acid, to sterilize instruments and wounds of patients. So that is why he is known as the father of antiseptic surgery. Understood? So this can actually come as an MCQ. Who is known as the father of antiseptic surgery? It is Joseph Lister. How do you remember it? SEP for septic, right? So he gave an antiseptic, that means carbolic acid, and he was using this carbolic acid to sterilize the instruments and wound of patients. So because of him, a lot of infections that was happening in patients decreased. Understood? Okay, I'm seeing a lot of comments. Yes, so everybody's giving right answers. Very happy about it. Good. So next scientist that we are going to come is Paul Ehrlich. So he is actually also a very, very important scientist because he also gave us a lot of theories and he also gave us a lot of uh, different organisms also. So how do we remember him? So Paul Ehrlich, right from his name only, we will try to have some mnemonic. So the mnemonic comes from CH, Paul Ehrlich, CH. What did he do with the CH? There were two things that he gave us with the CH. He actually gave us a drug, or actually he gave us the concept of chemotherapy. Right. So what happened? Actually, he gave us a drug called as salvarsan at that point of time, which was used to treat syphilis. So he gave us the concept of chemotherapy. He gave us the concept of chemotherapy, right? So that is why. What is the other CH? He, that is why, is known as the father of chemotherapy. Because he gave us the concept of chemotherapy, that is why he is known as the father of chemotherapy, right? There was another CH that he gave us. What was that? That was the chain theory for antibody production, right? So these are the three CH that he gave us. He gave us the chemotherapy, which was a salvarsan, which was a drug which was used to treat syphilis. Then that is why he is known as the father of chemotherapy. And he gave us another concept with the CH, which is known as the chain theory for antibody production, right? So that is one thing from his name. Let's again write his name, Paul Ehrlich, right? What is the other thing that he gave us? So from his name, he actually gave us the concept of acid fast nature of the TB bacilli. So he actually told us that TB bacilli is actually acid fast, right? That is the concept that he gave us. And what is acid fast? How do we do acid fastness? How do we check the acid fastness of TB? We do it by some staining, right? So he actually gave us another concept of staining techniques for tissues, right? He gave us the staining techniques for tissues and blood cells. Correct? Understood? Is everybody able to understand me? Right? Can everybody hear me? Good. So, next we come to the name Eldridge. So, what did he do? He gave us three things from Eldridge. One, he actually, just like the Pasteur Institute, he also made his own institute. So, he made a Paul Ehrlich Institute in Germany, right? So he made a Paul Ehrlich Institute in Germany. That was his first achievement. Then, with the Ehrlich, what did he do? He actually discovered a bacteria, and that bacteria was named after him, called as the Ehrlichia bacteria, right? So that is the second thing. And the third thing that he did was give us the phenomena which is called as the Ehrlich phenomena. So what is the Ehrlich phenomena? He actually gave us the concept that whenever a toxin will be formed, some form of antitoxin has to be formed which will neutralize that toxin. So he gave us the toxin antitoxin reaction phenomena. Got it? Understood, everybody? Great. So let's revise this again. Paul Ehrlich, CH. What did he do? He did three things with CH. He gave us the concept of chemotherapy. That is, what chemotherapy did he do? He actually invented salvarsan, which was used to treat syphilis, right? So that is the first thing. What is the second CH? He is known as the father of chemotherapy, right? The another CH that you see. Then the third thing is chain theory for antibody production, right? Then the name Paul Ehrlich. Paul से A से क्या आया? Acid fast nature of the TB bacilli. Acid fast nature of the TB bacilli. And how do we check the acid fastness? We check it by certain staining techniques. So he gave us the concept of staining techniques for tissues and blood cells, right? Then come to Ehrlich. What did he do? He formed the Paul Ehrlich Institute in Germany, right? And then he discovered a bacteria called as the Ehrlich bacteria. He named it after himself, Ehrlich bacteria or Ehrlichia, right? And then he gave us the concept of Ehrlich phenomena, which is a toxin-antitoxin reaction phenomena. So this was all about Paul Ehrlich, right? Next we come to Robert Koch, a very, 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 very important scientist, which comes as a short note during your undergraduate sessions, and a lot of MCQs come from it because he has done a lot for the field of microbiology, right? So, is everybody clear up till now? Then only I'm going to move forward. I'm going to wait for you to give me some comments in the comment section so I know that you've understood from here. Is everybody? Yes, okay. So I'm going to continue. So the next scientist we are going to talk about is the Robert Koch. So I've actually named him as Robert Koch. Can you see? I've actually named him like this. Why? Because I'm going to write S and I'm going to write CH again from his name only, right? So S, what did he do? He actually gave us the concept of solid media using agar, right? He gave us the concept of solid media using agar. So what used to happen before is that a lot of natural things were used initially to grow any kind of bacteria or funguses or something, and or liquid medias were used. But he actually used agar, which is kind of a weed which used to be used to solidify things. So he actually got the concept and he used agar in a way that he used it to solidify a media and grow things on that media, right? So he gave us the concept of solid media using an agar. So that is the S. What is the CH? He gave us the concept of culture in pure form, right? He gave us the concept of pure culture. That means an organism can be grown on the media in a pure form. That is known as pure culture. And H, very good. People are already giving me, answers in the comment section. Very happy about it. H is for the hanging rock. Yes, H is for the hanging drop, which is used for the motility of an organism, right? It is used for the motility of an organism. So I think a lot of people are doing these practicals in your undergraduate microbiology practicals where you're using hanging drop as one of the methods to check motility of an organism, right? So who gave this concept? Who gave this concept? Robert Koch gave this concept of hanging drop. So the three SCH, Robert Koch ka S, C and H. S is for the solid media using agar. C is for the culture in the pure form. And H is for the hanging drop used for the motility. Right? Then what did Koch do? Remember, Koch had a cat. Right? Koch had a cat. So what did he do? He actually discovered three bacterias. He actually discovered three bacterias. Can you tell me which all bacterias did he discover? The cholera, right? The Vibrio cholerae, the anthrax, and the most important one and the most prevalent one here in India, the TB bacilli, also known as the Koch's disease, right? Also known as the Koch's disease. It comes very, very commonly. This question, Koch's disease, what is it? It's actually the TB bacilli or the tuberculosis, right? So these are the three bacteria that Koch actually discovered. So Koch had a cat, and cat has CAT, cholera, anthrax, and TB, right? Then what else did Koch do? He gave us few phenomena. So the first one is known as the Koch's phenomenon. So what was the Koch's phenomena? Can anybody tell me? Okay. So Koch's phenomena is actually, he said that if he takes this TB bacilli and injects it into a guinea pig, right, that guinea pig will have a hypersensitivity reaction towards this TB bacilli. That is known as a Koch's phenomena. So that means TB bacilli, if injected in a guinea pig, it will have a hypersensitivity reaction. Got it? So this is the Cox phenomena. Understood? Great. Now let's come to the most important thing that he did. He gave us the concept of Cox postulates. Cox postulates, right? So what did he say actually? So he gave us five criterias. He gave us five criteria. So what are those criteria? I'm going to actually try and uh, draw this for you if you can understand. So, okay. So let's see. I'm trying to draw a mouse, right? Can you see? It's a mouse, right? So let's see. It's a mouse. Okay. So the first thing that it said was that whenever an animal is injected with some disease or is infected with a disease, the organism should be constantly associated with the disease, right? So, if some organism infects the mouse, okay, I'm going to zoom it in. So, if an organism infects the mouse, that organism should be constantly associated with the disease. That means at every point of time, that organism should be present within that animal, right? So that is the first one. Then what did he say? Suppose it caused some kind of an ulcer on the mouse, some kind of a hypersensitivity reaction, right? Sorry. So next what he said is that if I take some sample from this wound and I grow it on a plate, right? If I grow it on a plate, I should be able to isolate this in pure form. So isolate organism in pure form. So that was the second postulate that he gave us, that taken an organism or taken the sample from the mouse and I'm growing it on a culture plate. So I'm able to isolate the organism in pure form, right? Then what did he do? Then he said, if I take these colonies or this organism and I inject it in a mouse again, okay, drawing a mouse again, right? So if I take this organism and inject it in the mouse again, it should cause the same disease, right? So inject in another animal, cause the same disease, right? That is the third postulate that they gave us. Then what? Then he said, if I take this sample again from this and now I'm again isolating it in a culture plate, right? What should happen? Again, it should grow in pure form. Again, it should be able to isolate in pure form. Understood? So these were the four postulates. Then what he said, if from this, if from this mouse only, I take serum of this mouse, what is this serum going to show me? Is everybody able to hear me? Everybody able to hear me? Great. Okay. So if I take serum from this mouse, right, and I check for the antibodies, I should have specific antibodies against that organism, right? I should have specific antibodies against that organism that will protect the host, right? So this is actually the fifth postulate, which was added later. Initially, there were only four postulates, and this fifth one was added later, right? So again, we're going to revise the Koch's postulates. What are the Koch's postulates? First, if an organism should be constantly associated with the disease when infecting an animal or a human or any person, right? If it's producing a lesion, I'm taking a sample from this person, injecting it on a culture plate. It should grow. This organism should be isolated in pure form, right? Should be isolated in pure form. Then from this culture plate, this organism, I'm injecting it into another, right? What is going to happen? It will cause the same disease, causing the same disease. Now, again, from that lesion, that disease that has been formed, again, from that lesion, if I take it and grow it on a culture plate, I will be able to isolate it in pure form, right? And then the fifth postulate that was added is that if I take serum from this infected person, what is going to happen? I'm going to check for the antibodies, and they should have specific antibodies against that organism that will protect the host, right? So these are actually the five postulates that Robert Koch gave us, right? And we were believing his Koch's postulates up till in the end, till few scientists came up actually, and they said that no, it's not about the organism, right? So what did they say? Okay, so there were few scientists who came and said it's not about the organism. Just the mere presence of an organism does not mean that it will cause a disease, right? So there was a concept, there was a new concept that was given, and that concept is known as modified God's postulates, right? Then they gave us the concept of modified Koch's postulates. So, what is the concept of modified Koch's postulates? So, few scientists came and they said that it's not merely the presence of organism, but some virulent gene that the organism possesses. It's said that it is about some virulent gene. It is not just the mere presence of the organism, right? So, this was one concept. Then they told us, okay, I'm going to draw like, okay, let me draw a circle. Okay, so let's say that it is a virulent, sorry, just a second. It is a virulent gene. So what did they say? They actually said that this virulent gene, so now they have given a certain modified Cox postulate. So they said that these virulent genes are actually associated more with pathogenic strains, right, than the non-pathogenic strains. That means the pathogenic strains will actually contain these virulent genes compared to the non-pathogenic ones, the ones that are not causing a lot of disease. So this virulent gene has to be present in the pathogenic strain to cause the disease. So this was the first one, modified postulate that they gave us. Then they said that if there is some level of mutation if some mutation takes place in this virulent gene. So what is going to happen? It is going to decrease the pathogenicity. That means it's going to decrease the amount of disease that it is going to produce in the person. So decrease the disease in the person, right? Can everybody hear me? Yeah. Okay. So that is the second one that they said. Then they said that if this mutated gene was replaced with the wild type is the one that's containing the virulent gene, right? With the wild type gene, then it will increase or restore the pathogenicity, right? That if I replace it with the wild type, it will restore the pathogenicity of this organism, of this gene. Then they gave us the concept, another postulate, which said that this virulent gene has to be expressed at some point during the infection. Right, at some point during the infection, this virulent gene needs to be present. What does that mean? So let me tell you, give you an example. Suppose a person is infected with tuberculosis only. So there is a time when it is in the latent period when it is not causing any kind of disease, right? But when the immunity of the person goes down, right? When the immunity of the person goes down, what happens at that time? This virulent gene gets expressed, and because this gene gets expressed, the person starts having clinical features and starts producing the disease. So this is what it means, that this virulent gene has to be expressed at some point during the infection cycle. Got it? And the last one, it says that the antibodies that are produced should protect the host, as that we said from the previous one only. That is the modified postulate also. Okay, that antibodies, here it said the antibodies are to be detected, and here it should protect the host. Got it? Now I want you to remember one thing, which is the exceptions of Koch's postulates. This is very, very important, the exceptions of Koch's postulates, because this can come as an MCQ. This comes as an MCQ very, very commonly. So, what is that time when the Koch's postulate can have an exception, right? So, the time is either we don't have an animal model or we don't have a culture, right? Either we don't have an animal model or we don't have a culture. So, these are the two times. So, no media or no animal model, right? So there is no media, that means it's empty, right? No media is empty. That means there is no media, the media is empty. So what is it? So Mycobacterium leprae, right? And leprae is the one, the first one, and the second one is T. pallidum, Treponema pallidum, which causes syphilis. So we don't have a media to grow these two organisms, right? So that is why this comes as an exception of the Koch's postulate. Not of the modified Koch's postulate, just the Koch's postulate. Please remember this, okay? And there is no animal model. That means we don't have an animal to inject this organism into. We don't know in which animal it will grow. So which one is that? Yes, people are giving me the right answers. It is Neisseria, which causes your gonorrhea or meningitis, right? So this is very, very important. Please remember this. The exceptions of Koch's postulates, very, very important. Please don't forget this. It can come as an MCQ in your entrance examination as well. Okay, so that is all about the scientist. Just one quick revision we can do here, right? Starting from the first one, Anthony Phillips von Leeuwenhoek, long name, long hair. That is how we're going to remember him. So he has Phillips in his name. Phillips makes microscopes, so he invented the microscope and he saw small animals in it and called them little animalcules. And that is why he is known as the father of microbiology because he invented the microscopes. And we can't see anything without the help of microscopes in our microbiology. Louis Pasteur from the Pasteur Institute, father of modern microbiology because he was doing a lot of modern research there. LNP, L for the liquid media, P for the pasteurization or the sterilization of milk. And he used to drive a G-Wagon car, giving us the three vaccines, the first one being cholera, then anthrax, and then your rabies vaccine. And from G, he gave us the concept of germ theory of the disease, right? This is how Louis Pasteur looks. Please remember him. Then comes Joseph Lister, just gave us one concept, SEP, septic, gave us the concept of an antiseptic, which is the carbolic acid, and he used it to sterilize the instruments and wounds of the patient, and that is why he's known as the father of antiseptic surgery, right? Coming back to Paul Ehrlich, CH, he gave us three CH concept of chemotherapy, which was the salvarsan used for the treatment of syphilis. Then the second CH, which is your father of chemotherapy, and the third CH, which is the chain theory for the antibody production, right? Coming back to his name, Paul Ehrlich, P ka A is for the acid-fast nature of the bacilli, the TB bacilli, and acid-fast is a staining process that we use. So he gave us the staining techniques for tissues and blood cells. Paul Ehrlich also gave us the Ehrlich Institute of Germany, discovered a bacteria called Ehrlichia, and gave us the Ehrlich phenomena, which is the toxin-antitoxin reaction. Coming to Robert Koch, Robert Koch had a cat, right? S is for the solid media, C for the culture in pure form, and hanging dog, which is your motility. Then gave us three bacteria, the cholera, anthrax, and the TB, which is also known as the Koch's disease. Koch's phenomena, which is the TB bacilli injecting into a guinea pig, then gave us the five concepts of the Koch's postulates, the four, and the later first one, the last one was added, which is the antibodies one. And then there were certain modified Koch's phenomena, which told us about the virulent gene, that it is not merely the presence of an organism, but the virulent gene, which codes for the organism, which codes for the virulence of the organism, right? So finally, the exceptions of the Cox postulate, which is your either no media or no animal model, right? So M. leprae and Treponema pallidum, no media, and Neisseria, no animal model, right? So the lastly, we are going to come to this MCQ. I want you to match the photo of the scientist with the name of the scientist. So that is how we remembered it. Can you do this? Match the photo of the scientist with the name of the scientist. Can you do that? Can you see these four names? Yes. So I want you to give me comments in the comment section and tell me. I'm waiting. Yes. So how did we remember it? Long hair, right? We remembered it as long hair, long name, right? Then we remember it as this is Robert Koch, right? The one that has a side look is Sir Paul Ehrlich, and this is Louis Pasteur. So our answer becomes A1, B3, C4, and D2. Is it right? So people gave me Cox, then Pasteur, then Leeuwenhoek, and then Paul Ehrlich. Very good. So This concludes our class for today. So if you have any doubts, please write it down in the comment section so I can take up the doubts if there are any. The rest, I think I've tried to summarize this concept as much as possible, and I hope it helps. So, yeah, any more? So, I think we can stop this now, and I will be taking up more sessions like these, the live sessions of further general microbiology. So, please be there for the classes. Yeah, thank you.